All right. We are starting into our unit about the cardiovascular system and the lymphatic system. This will relate to the immune system, where we are looking at things that are related to infectious diseases and how our immune system reacts to those. We'll start with just briefly going through chapter 12, which will introduce us to white blood cells in the body. And then we'll move into the lymphatic system where we'll learn about the different types of immunity and cells that are associated with your immune system. So to kind of start with, blood is considered a connective tissue. This connective tissue is consistent of cells, namely red blood cells, white blood cells, and platelets that are considered what we call formed elements, and they are floating in a fluid matrix, a liquid matrix, which is blood plasma. Okay. We can actually take a, centrifuge tube of blood, spin it around really quickly using centripetal force, and we can actually separate out each of these. So the formed elements are the cells themselves, red blood cells, white blood cells, and platelets, which are fragments of cells. And then we can have that separate liquid portion, which is your blood plasma. It gives you kind of a quick definition here, although we are going to go into the types. So red blood cells, their main purpose is to transport respiratory gases. This would be oxygen and carbon dioxide around the body. White blood cells are a part of our immune system for fighting infection. There are multiple different types of white blood cells, which we will be learning about, and they all have their own separate function. Platelets are pieces of cells, fragments of cells called megakaryocytes, and they are part of blood clotting for stopping bleeding. And then plasma has a lot of different proteins in it, proteins that are good for people, such as antibodies, which is why sometimes people donate blood plasma. This is kind of what this looks like. So if we take a blood sample, 45% of that sample will be the cells themselves, which are called the formed elements, and 55% of that sample will be the liquid matrix, which is plasma. You can see there's a lot of components to plasma. You are not going to have to memorize those, okay? And then you can see here that for the formed elements, most of a blood sample is red blood cells. About 5% are platelets, those fragments, and then a very small percentage are our white blood cells, but we are going to focus on our white blood cells, these different types that you see down here in detail. So before we kind of get into that, this is just showing just the same things that that figure showed before, okay, just in case you want to kind of see that in less of a infographic, but more in written word. Okay, so showing us all these normal things. This is just a throwback to the urinary system, right? Where we did learn about the blood's pH needs to be within this very specific range of 7.35 to 7.45 on the pH scale. And, part of that maintenance, we have all of these formed elements. There is a specific name for the red blood cells you saw before on this figure. Red blood cells are most of the sample for formed elements in a drop of blood, okay? And when we do spin them around in centrifuge tubes and we get the different components, red blood cells are termed hematocrit, HCT. And again, plasma makes up 55%. We have a small percentage that is white blood cells, but we'll take a look at that still. This is that centrifuge tube that I'm talking about, the substance in here. So all these tubes are coated so that the blood does not clot, because you cannot centrifuge out all these individual elements if the blood clots. So that's why we've got different chemicals found in here. These tubes are called heparinized tubes because heparin prevents blood clotting. Right, this machine here is a centrifuge. It's going to use centripetal force to spin that sample in this centrifuge tube, and then we get these separate elements, okay? This would be what we would call a blood smear, something you could look at under the microscope after you stain these cells. Most of the sample is red blood cells. We have a couple fragments for platelets, and then we have white blood cells here. This is what the sample would look like just coming out of the centrifuge tube. Plasma would be the least dense, so it would be at the top. This buffy coat in here, you will actually see a physical kind of thin line there that looks fairly white, and that will contain the white blood cells and platelets. And then the bottom of the sample is the hematocrit, the red blood cells. So we'll start with red blood cells. We'll look at each formed element, although we are going to focus on the white blood cells. So red blood cells, that is their common name, but their scientific name is erythrocytes. So red blood cells are called erythrocytes. And if you remember, their main purpose is to carry respiratory gases around the body. Their structure is these biconcave discs. They look like SpaghettiOs almost, okay, except there's no hole in the middle. And that provides surface area for gases to attach. Those gases attach to the red blood cells through a protein called hemoglobin. And really, when we talk about red blood cells and what they're made of, oftentimes red blood cells are referred to as bags of hemoglobin. There is a large amount of hemoglobin on these red blood cells, allowing oxygen and carbon dioxide to attach. When oxygen is attached, the hemoglobin is called oxyhemoglobin. When carbon dioxide is attached or when oxygen is released, we call it deoxyhemoglobin. This is that biconcave structure. And red blood cells are some of the smallest cells in the body by far. You can see the 7.5 microns. This is just the structure of hemoglobin. It is a complex protein structure that has these heme groups, okay? The heme groups in the center here that you can see in red, they have an iron center. That's what Fe is, and this allows us to bind oxygen and carbon dioxide. Typically for red blood cell counts, and this will be different for male versus female. In adult males, we have, a decent amount of cells, okay, per millimeter squared, and it's slightly less for adult females. I don't need you to memorize these numbers. And the amount of red blood cells that you have and the ones that have oxygen bound to them, that oxyhemoglobin, can kind of tell you your blood's oxygen carrying capacity. And if that carrying capacity is low for some reason, this could be indicative that you have some sort of condition or disease. Before we get into those diseases, which I think we will have eventually, and for red blood cells, the disease that we may get to at the end of this PowerPoint is anemias. But for red blood cells, if there is a low oxygen carrying capacity for some reason, maybe there's not enough red blood cells, or maybe there's something wrong with the red blood cells to where they're not carrying oxygen or they're not in an environment with oxygen, your body will be stimulated to produce more. And that process is called erythropoiesis when we make more red blood cells. And again, red blood cells are called erythrocytes. So erythropoiesis is making more of them. So we learned before that the function of our red bone marrow is hematopoiesis, which is just general blood cell formation. But if we want to do erythropoiesis, this is just part of that process, except we're specifically focusing on when red blood cells are made. So any of the cells that we talk about, we'll see the chart in a minute, start with some sort of stem cell. For blood cells, we call them hemocytoblast or hematopoietic stem cells. And eventually, different factors, chemicals, are going to act on these stem cells, and they're going to become our different types of cells, one of which is going to be the red blood cell. So the chemical hormone that is acting on these hematopoietic stem cells to make them make red blood cells is erythropoietin. Again, has that erythro name in it for the erythrocytes that it's creating. We can see that process here. Here's the stem cell. It can differentiate into two different types of stem cells. One is lymphoid, and the other is myeloid. Myeloid gives rise to most of the other cells. And then we can see here, if we had the hormone erythropoietin activating this myeloid stem cell, we are eventually going to form our erythrocytes, our red blood cells. Let's move on to white blood cells. There are many different types. Our white blood cells scientifically are going to be called leukocytes. That prefix leuko means white. They, again, also rise from those hematopoietic stem cells, hemocytoblasts. When they are given rise to, there are a number of different ones that can form, and they do different motions. In general, this list is going to be what a lot of white blood cells do in general before we get into the types. One thing that many different types of white blood cells can do is a process called diapedesis. Diapedesis is when these white blood cells are circulating in the bloodstream in blood plasma, but then they stop and attach to the wall of the blood vessel, and then they go in between the capillary junctions to get into the tissue. So they leave the blood vessel to get into the tissue. That is called diapedesis. There's other things that can happen. So once a white blood cell leaves circulation, leaves the blood vessels, it can do amoeboid motion, okay? Meaning it acts kind of like an amoeba, which is a small single-celled organism, right? That can move kind of in a swimming-like motion. They also do something called positive chemotaxis. Chemotaxis means that they're attracted to some sort of chemical that the cells are giving off. When cells become damaged or infected, they will give off signals, and even the bacteria or virus that might be infecting them will give off signals. And this will kind of act as a trail of breadcrumbs so that the white blood cell can make its way over to that area. That's called positive chemotaxis. Just like we saw with the red blood cells where they needed erythropoietin to stimulate their production, when we are doing this process from the myeloid stem cell and we want to make all of these white blood cells, we have other chemicals and they are called interleukins and colony stimulating factors. Those are going to be the ones that act on the stem cell to make it differentiate into a white blood cell. This is just what diapedesis looks like. It's actually a fairly good graphic, right? So we have a leukocyte, general white blood cell, squeezing between the lines of these capillaries, right, to go into tissue, diapedesis. So let's get into the 5 types. So not only are there 5 different types of white blood cells, but they're separated into 2 categories. 3 of them fall into the category of granulocytes, and these are white blood cells that have granules inside of them. So they have what we would call granular cytoplasm. You can physically see granules inside their cytoplasm. The 3 types of granulocytes all end in "-phil", if that helps. Okay, so neutrophil, eosinophil, and basophil. Neutrophils, these ones circulate pretty widely. They have the strongest circulation. We call them strong phagocytes. What that means is that when they go to an area where they need to take care of an infection or injury, they're going to do phagocytosis. They're going to engulf whatever should not be there, process it, and get rid of it. So they're strong phagocytes. And essentially, these guys are actually the first to arrive at the infection site. So as soon as you become infected or injured, neutrophils are going to be the first ones to come to that area and they're going to start phagocytosis. These other 2 are less commonly circulating in the blood, eosinophils and basophils, and they only show up in very special situations. Eosinophils, they only kind of really appear when there is parasitic infections or inflammation occurring, where you might see them appear in a blood smear. Basophils, they are ones that really show up in response to allergies, allergic reactions, and they're going to actually release specific chemicals that kind of help with that allergic reaction, promoting inflammation. They release heparin and histamine. Remember, heparin is something that causes blood cells to not clot, keeps blood flowing to that area, and histamine is highly inflammatory, right? Causing a huge inflammation response needed to, basically create positive chemotaxis so other cells come to that area and take care of the problem. This is why sometimes when we have allergic reactions to things, we take antihistamines to kind of reduce that inflammation. This is a neutrophil. This is just what its appearance looks like. It is characterized, you can see those granules, so you know it's a granulocyte. It's surrounded by red blood cells, and it is characterized by these multi-lobed nuclei. So this is one nucleus, but it has 4 lobes. Eosinophils are very characteristic. They not only have these multi-lobed nuclei, but they also stain red. Okay, and you can see all these granules staining red. Last of our granulocytes is the basophil. Basophils also have these multi-lobed nuclei, but the granules inside of them are very large, and when they stain, they almost obscure the nucleus. This is what this basophil looks like. And again, the basophil one is the one that releases heparin and histamine in response to kind of call other cells to the area. Our last 2 types of white blood cells are considered A granulocytes, A meaning not or none, so they don't have granules, and they stay in the bloodstream for a very long period of time. Um, one of the biggest ones is called a monocyte, and we'll see pictures on the next slide. It has a kind of bean-shaped large nucleus. And these are also strong phagocytes. So they do phagocytosis just like neutrophils. But one of the things that monocytes like to do is migrate to tissues, okay? And that migration, right, we leave the bloodstream and go into tissues is called diapedesis. So these guys like to do diapedesis. And when they go into tissue, they actually become a cell called a macrophage, where they just do phagocytosis and try to get rid of whatever is in the tissue that shouldn't be there. The other type of agranulocyte are lymphocytes, which we'll be talking about in detail for the next chapter. Lymphocytes are the smallest ones. They have a large nucleus that basically covers the whole cell. They're very tiny, and they are the ones that are going to help us produce our immune responses. Lymphocytes are T cells and B cells and natural killer cells, abbreviated here. We'll learn more about these, but B cells are the ones that eventually differentiate to produce antibodies. This is our monocyte, large, very large cell, right, compared to these red blood cells, and has that kidney bean shape nucleus. This is a lymphocyte, almost as small as the red blood cells, and it has a huge nucleus that takes up most of its cytoplasm. As far as white blood cell counts go, there is some general range of white blood cells, and obviously that range will get higher if you are have inflammation or infection. If your white blood cell count goes up high, this could be indicative of an acute infection, so a recent, infection, ongoing infection. And when your white blood cells get higher, it's called leukocytosis. Leukocytosis also could be associated with certain blood cancers like leukemia. If you have a low white blood cell count, this could be due to certain types of viral infections where the virus actually infects white blood cells and kills them. This could include AIDS, chickenpox, typhoid fever, measles, mumps, all these nasty things. Then you could have a low white blood cell count characteristic of a disease, and that could be called leukopenia. A lot of times with the flu, influenza, if someone has a particularly bad case, a good sign is this leukopenia, low white blood cell count. And typically, when blood samples are taken and we want to look at what percentage of each type of white blood cell is there, we call it a differential white blood cell count, where they can look at the relative percentage of each type. So say you were infected with some kind of worm, you had some kind of gastrointestinal parasite, you might look at that differential white blood cell count and see that you have a high eosinophil count, because eosinophils are going to be present during parasitic worm infections. The last type of formed element is blood platelets. These are also called thrombocytes. And for these guys, they're actually not cells at all. They're fragments of cells. And the cell that they are a fragment of are called megakaryocytes. Megakaryocytes live in the bone marrow, but when they try to exit the bone, they break apart into fragments. So these fragments are called platelets or thrombocytes, and the stimulation for us to make these megakaryocytes, that hormone is thrombopoietin. And their main purpose is to stop bleeding, cause blood clots, and that process is called hemostasis for creating that blood clot. Last kind of thing for components of blood, this is the liquid portion, plasma. It generally is a yellowish color. It is mostly water, but it contains a lot of different substances, including proteins, one of which is antibodies. So for our plasma proteins, these are the three groupings. We have albumins, globulins, and fibrinogen. Globulins is where we're going to find our immunoglobulins, which is antibodies. So speaking of antibodies, and we'll kind of get a little bit into how they form, okay? When something enters the bloodstream that should not be there, okay, it will evoke an immune response. And that molecule that the immune response is tailored to that will evoke it is called an antigen. We have antigens on our cells in our body, and those are called self-antigens that tell us, hey, this is me. But when something new is introduced, so a bacteria, a virus, even an organ transplant, if it's not yours, it has antigens on its surface that will tell your body, this is foreign. We need to take care of this. So an antigen is a molecule that evokes that response where the body knows that it's not part of it. And when that antigen evokes a response, antibodies can be formed. And these are a type of plasma protein that will specifically attack an antigen that whatever it makes it towards. And usually it's going to be an antigen that's not a person's own cell's antigen. It's going to be a foreign one. There are a few reactions that can occur. One of the ones that's very common is transfusion reactions. Okay. This is referring to blood type, and we're not going to get into blood type too much into this class, but the reason why you cannot accept a blood type from another person is because our blood cells contain antigens. And if you're going to receive a blood transfusion, you need to receive the same type of blood that you have, not something that is opposite that might evoke an immune response. So I am type O blood, and the best thing for me to receive is type O blood, right? There are some exceptions to this, okay? I'm following the ABO group. We're not going to get into that in this class. But if there is some sort of transfusion reaction, so say a patient in the hospital was given the wrong blood type, primarily what's going to happen is antibodies are going to form and they're going to do something called agglutination. And what agglutination does is it basically causes the red blood cells to all clump together. So instead of them circulating individually and nicely through the bloodstream, through blood vessels and blood plasma, this antibody is going to cause agglutination where those red blood cells that are not correct, right, from that transfusion make all the blood cells stick together. This can form blood clots. This can cause poor circulation, lots of problems for the person receiving that transfusion. And this is kind of what that looks like, right? We might have cells kind of free and about, but if those antibodies, right, are going to find it, it can cause this clumping, and that can cause a lot of problems in a patient who is probably receiving a blood transfusion because they really need the blood. And this is essentially, if you give them the wrong blood type, making it useless. That is the end of chapter 12. Okay. We have one more chapter for the lymphatic system, which I'm going to go to in just a moment. Let me share my screen for our last chapter in this unit. All right. So we just went over blood in chapter 12. Chapter 14 is the lymphatic system, also referred to as the immune system. So the lymphatic system is considered your second circulatory system. We would consider the first one your blood, okay, circulating through your body in those blood vessels being pumped through by your heart. Next to your blood vessels, you also have other vessels called lymphatic vessels, and they are circulating not blood, but lymph. And this is very important for your immune system as well as fluid movement throughout the body. What these lymphatic vessels do, and they can be very similar size and orientation as blood vessels, they collect and carry away excess tissue fluid. So a lot of times we have fluid from the blood that can make its way into the tissue. And if it hangs around for a while, sometimes we can see edema or swelling, right? But that swelling usually goes down or goes away, and that's because these lymphatic vessels are going to take away that excess fluid. Additionally, with these lymphatic vessels, there are specialized ones that absorb specifically fat and help carry fat throughout the body and introduce it back into the circulatory system. There are different structures that lymphatic vessels attach to, which will filter lymph as it goes through the body. One very important one are lymph nodes. Okay, we know when we get sick, sometimes our lymph nodes feel painful, swollen, that type of thing. It is because in your lymph, they are finding bacteria or viruses. It's circulating in that lymph and it's made its way to a lymph node, which is going to try to take care of that infection before it goes to the rest of your body. This is kind of what this looks like. So eventually this lymph will make its way back into your blood system, but it's in a separate system. And kind of the main filters, making sure that we don't get sick and introduce bacteria, viruses, or whatever into the bloodstream. We have these lymph nodes here, which are going to filter out all that lymph to make sure that there's no infection spreading throughout the body. We know that if infection is able to spread to the bloodstream, this could make a person go into septicemia. So kind of the function of these lymph nodes, they filter the lymph, removing bacteria, cellular debris before it goes to the blood. They do immune surveillance. That means that if they do encounter a bacteria, a virus, or something else, they can actually start to mount a defense against that because they know that there's an infection somewhere in the body, okay, that they've encountered. So they do this immune surveillance. They start producing lymphocytes and macrophages. Remember, these are our agranulocytes. Lymphocytes are T cells and B cells, which we'll talk about. Macrophages are what white blood cells, those monocytes, become when they enter tissue. So those lymph nodes are going to be kind of the center of lymphocyte production, making lots of those T cells and B cells. These cells can attack viruses, bacteria, other disease cells, okay, that come into the lymph node. These macrophages can actually leave and go and engulf and destroy foreign particles, debris, and other damaged cells in the body. So this is really at the lymph nodes where we're mounting a defense. And if your lymph nodes are swollen or painful, likely there is an infection, right, that they are trying to take care of. Another structure of importance for the immune system is your thymus, okay? Your thymus is located right around where your heart is, okay? A little bit above, right behind the sternum, okay? And your thymus is actually the place where your lymphocytes can mature into T lymphocytes or T cells. That's where that T comes from, means thymus, okay? So T cells mature at the thymus. That's where the majority of those mature ones that can recognize specific antigens are going to come from. What stimulates them to become mature is a specific hormone called thymosins. So we have those lymph nodes that are producing lymphocytes, right? Immature ones, and producing macrophages. Those immature lymphocytes are going to eventually go to the thymus, okay, some of them, to become T cells, and they mature through hormone action, thymosins. Here's your thymus right here, right around the heart, behind the sternum. And this is just going to be different areas where your T cells are going to be formed. T cells are a very specific type of lymphocyte which can recognize specific antigens so that they can destroy infection or infected cells. So what we've really been talking about that are producing antigens, okay, that the body can recognize are pathogens. And pathogens is a very generalized term for disease-causing agents. This can include bacteria, viruses, fungi, or protozoa. There are two types of defenses that your body is going to have when a pathogen enters your system and causes infection. First, what's going to happen is what we call our innate defenses. These are considered non-specific. So your body recognizes that something foreign is there, but it doesn't recognize any kind of specific antigen. It just knows that this is not supposed to be here. There are a lot of innate responses that are going to happen right away, responding quickly as soon as infection happens. These include species resistance, mechanical barriers, chemical barriers, natural killer cell formation, inflammation, phagocytosis, and fever, which we'll talk about. After the innate defenses have started, eventually your body, as those pathogens circulate in your lymph nodes, eventually your body is going to be able to actually recognize what is infecting it, what it specifically is. And that's when your adaptive or specific immunity comes in place. It's going to respond more slowly because it's going to take it a little bit to recognize the specific type of pathogen. But once it does, it's going to create specialized lymphocytes, T cells and B cells. And those B cells are going to produce antibodies. That's where that specific defense comes from, but it takes a little while to ramp up. So let's look at those innate non-specific defenses first. One is species resistance. And simply what that means is that if there is a pathogen infecting one species, a lot of times it cannot jump to the other species, okay? There is examples where this does happen for sure, right? We've heard of like bird flu, swine flu, things like that. COVID, right, can jump from animals to humans. But generally, there are other pathogens that simply can't cross the species barrier, right? If they tried to go from, like, say, a frog, fungus, can't really infect me because I don't have the right receptors, I don't have the right body temperature, things like that. And we would just call that a species barrier, species resistance. Another innate defense is mechanical barriers. Mechanical barriers are just physical barriers that will separate the pathogen's ability to enter the body. Your first mechanical barrier is your skin. After that, we've got your hair, mucus production, sweat. All of these are trying to prevent the entry of the pathogen physically into your body. Okay, so it's kind of our first line of defense, is this mechanical barrier. We're going to look at our second line of defense, which is all these other innate defenses coming after this. If the pathogen makes it past the mechanical barrier, the first line of defense, say you got a cut, right? That physical barrier is broken. Now we're going to go to our second line of defenses, the rest of these innate defenses. One of them is inflammation. We all know what it feels like to have inflammation, right? Think about maybe if you get a splinter in your hand, it's going to create kind of a hot, painful, swollen area. That is inflammation, characterized by these things: redness, swelling, heat, pain. And what this is essentially doing is it's trying to keep the infection in one place, or keep the injury in one place without spreading it to other parts of the body. Inflammation is keeping it in one area so that it can let those white blood cells come in and try to take care of that problem. Another defense, part of our second line of defense for innate defenses, is chemical barriers. There are lots of chemicals in your body that will kill pathogens. One example of this could be acidic environment. Certain pathogens, if you consume them, your hydrochloric acid in your stomach could kill them, right? So they don't become a problem. There are other organisms that have found a way to evade this, right? But certain pathogens can be killed by just simply an acidic environment, like the one in your stomach. There are different enzymes that you can produce. So when you cry, there is different enzymes in your tears, like lysozyme. This is meant to be antimicrobial, antibacterial, so that you don't get infections in your eye. That's why tear production can be pretty high if you get something in your eye. There are also other chemical barriers like interferons and complement. These are groups of protein or protein-like structures that are going to try to prevent viruses or other things from making more of themselves. Another innate defense, if the pathogen makes its way past your mechanical barrier, is natural killer cells. These are a type of lymphocyte, but they don't have any specific recognition of a pathogen. All they will do, and they mostly target viruses and cancer cells, is they will produce substances that kill cells, essentially cytolytic. They're called perforins, and they will release those perforins that attach to the cell membrane of viruses or cancer cells, and it kills the cell. These perforins, as you might imagine, can also create inflammation. Another second line of defense for innate defenses is phagocytosis. Before certain cells even learn to recognize a pathogen, because the only ones that really do adaptive immunity are lymphocytes, okay? T cells, B cells. But you have other white blood cells like neutrophils and monocytes, right? Remember, they're both good phagocytes. They will go around and they will do phagocytosis in order to remove, right, things that should not be there. That is an innate defense. I think this might be our last innate defense. Our last innate defense is fever. Okay. Typically, you have a set point in your body for what your body temperature should be. But in the case of infection, there might be signals, which are called pyrogens, which make their way to your brain and tell your brain to produce a fever to try to either stop a pathogen from reproducing or kill the pathogen. And what the fever is doing is making a hostile environment for that pathogen. Okay. Unfortunately, That hostile environment is also going to be hostile for your own cells. So that is why we cannot have a fever persist for hours and hours and hours, because it will start to hurt the rest of your cells, not just the pathogen. But fever, for an appropriate amount of time, is good for creating a hostile enough environment to kill pathogens or at least prevent them from reproducing quickly. Lastly, we'll move into adaptive specific defenses. This is going to involve lymphocytes. We term this our third line of defense. This is when we actually start producing cells that can recognize specific pathogens. And these cells, these lymphocytes, are put into two categories. One we call cellular immune response. This is going to involve T cells, okay, mostly. And then humoral immune response is going to involve antibodies that B cells make. So here's kind of that structure. This is a summary, okay, of what we've gone through so far. All right. And so we've got our antigens again, right? They're evoking an immune response, okay? And so when we find that specific antigen, there are going to be cells, these lymphocytes, which are going to learn to recognize it. When T cells learn to recognize it, they do this in the thymus. Okay, that's what they're called, T cells, T lymphocytes. And they're going to specifically learn what the antigen is, and then they're going to go out and circulate and go to different areas of the body so they can try to take care of that infection. Other cells are going to differentiate in the red bone marrow. Specifically, B cells do this. Okay, so we learned that our lymph nodes, like I mentioned before, are the sites for lymphocyte production. But those lymphocytes need to learn what the antigen is in different places. T cells learn and mature in the thymus. B cells learn and mature in the red bone marrow. All right, we've got these kind of different ones here. Right? Thymus, lymph node, bone marrow. Okay. So they have to be activated, okay, so that they can respond to specific antigens. And what activation just means is they need to be taught what the antigen is. T cell activation occurs due to these antigen presenting cells, which are called APCs. These are specific cells, okay, that have encountered the pathogen, eaten it, phagocytized it, and now they're showing it on their surface to teach the T cells, this is a bad antigen. You need to be able to recognize this. APCs. So lots of times macrophages in the tissue do this. Macrophages, again, are monocytes. They engulf things. Then they will display that antigen on their surface, antigen presenting cell. T cells interact with them, and then they learn what the specific antigen is. Part of this goes along with what we call major histocompatibility complexes, MHCs, where basically because we have these antigen presenting cells showing up different antigens, this will form kind of a profile for different pathogens so that T cells can recognize them. Our T cells, again, as mentioned, are part of the cellular immune response, where cells are specifically involved. T cells, they are going to directly interact with antigen bearing cells, so either infected cells or the pathogen itself. They secrete things like cytokines, which will help enhance their response. They also secrete toxins, growth inhibiting factors, or other things that are essentially will kill or prohibit the growth of whatever cell or pathogen is damaging. There's multiple types of T cells. Cytotoxic T cells are the main ones that are doing all of this cytotoxic activity, killing things, killing cells that are infected. Helper T cells are ones that are going to go and actually show the antigen to B cells to help them make antibodies. And then we have memory T cells, which kind of hang around for a while in case we need a quick response for future exposures to the same pathogen. They remember the antigen from previous infection. This is kind of what that looks like, okay, showing each different type. And we can see that whole process here. This is a macrophage, right? It's engulfing an antigen-bearing agent, okay, so an infected cell of some kind. It's showing it to the cytotoxic T cell. It's learning, okay? We make more cytotoxic T cells, right? We can make memory cells to hang around later. Those cytotoxic T cells, okay, can produce different kinds of things, right, to attack directly. And then some of them become helper T cells, which can teach B cells what the antigen is so that B cells can become activated and eventually produce antibodies against these specific antigens. The B cell itself, it can become activated and it essentially produces clones of itself. B cells are associated with the humoral immune response. Humoral means the humors of the body. So plasma in the bloodstream is considered a humor, a liquid of the body, and that is where antibodies are going to exist in the plasma. And again, these B cells become activated by helper T cells that show them the antigen. The ones that actually make antibodies are the B cells that differentiate into plasma cells. And once they become plasma cells, they produce and secrete antibodies, also called immunoglobulins. That's our specific type of plasma protein. And then they can circulate in the body as part of our humoral immune response. Some other B cells will differentiate into memory B cells and remain dormant for future encounters with the same antigen. This is kind of what that looks like. Antibodies. There are a number of antibodies, okay, but we put them into different type categories. The most abundant, excuse me, are going to be these first three, IgG, IgA, and IgM. Okay, Ig just stands for immunoglobulin, that plasma protein. For IgG, these are found in tissue fluid and plasma, okay, circulating the bloodstream. Again, defend against bacterial cells, viruses, toxins, and activate complement, one of our chemicals that's going to try to directly attack these things. IgA, not really found in the bloodstream or tissue fluid, but found in exocrine gland secretions. So we find them in things that secrete out to the skin surface, like breast milk, saliva, tears, nasal fluid, gastric juices, bile, urine. You can find levels of IgA. IgM, again, found in the plasma, so circulating in the blood. IgM can be present when food antigens or bacteria are present. A lot of times foodborne illnesses are from bacteria. IgM can even occur if you do a mismatched blood transfusion, so they could be implicit in that. IgD, okay, is found on B lymphocytes, so B cells, and help with B cell activation along with those helper T cells. And IgE, which is probably what most people are familiar with, it's found on basophils and mast cells. These are the ones that are produced during allergic reactions, because you remember basophils are the ones that release heparin and histamine, causing heavy inflammation, right? Might be the reason why we take antihistamines. IgE is associated with allergies, allergic reactions. There are kind of three ways that antibodies can react. They might directly attack through agglutination, right? That clumping, precipitation, neutralization. They can activate complement, right? So that we do a lot of chemical warfare things. Or they can stimulate local inflammation, calling other cells to that site. For our immune responses, typically, and this is going to be an adaptive immune response. So this is looking at what immunoglobulins, antibodies show up first in the body. In a primary immune response, typically you have IgM show up first, then IgG. You might have seen these different levels when back when COVID was first starting out, people were measuring how often you would have that immune response and you'd produce these specific antibodies, okay? Your primary immune response, again, is going to be when you first encounter the antigen, what that's going to look like. And typically, we don't start to ramp up these nice antibodies until 5 to 10 days after exposure. So it takes a long time for that adaptive response. But say you're exposed to the same antigen again later on, you will have a secondary immune response, which is much quicker, longer lasting, and you already have some of these memory cells in place so that your antibody production is going to be very quick, 1 to 2 days versus 5 to 10. This is kind of what that looks like. So this might be when you're first exposed to a pathogen. It takes a while. This is days, right, on this x-axis, and you can kind of see it takes, to mount a response 10 to 15 days, maybe even, right, to get a full response. But secondary re-exposure to the same one, much quicker, and you'll be able to feel better faster. So this goes down to the last couple concepts that we have here, and you will definitely need to know these for your quiz. This is naturally acquired versus artificially acquired immunity, and then active immunity versus passive immunity. I'm going to tell you these separately, but then they kind of mix match with each other because you can have naturally acquired active immunity, naturally acquired passive immunity, artificially acquired active immunity, and artificially acquired passive. So they do mix. Naturally would be due to natural events. So you encounter a pathogen in your environment, right? Artificially would be through an injection, namely a vaccine. Active immunity means that you form your own antibodies because you were exposed to the antigen itself. This is eliciting an immune response for your own body. Passive means that you're receiving antibodies from someone else. You did not come in contact with the pathogen. You didn't create an immune response, but you are receiving antibodies from someone else. So we can look at some examples of this, and this is the slide I would definitely memorize, okay? Naturally acquired active immunity. Natural means just natural exposure. Active means you elicit an immune response. We can think of this, so if we were physically in the classroom and I came over and I sneezed on someone and they got sick because I had a cold, that is naturally acquired active immunity. Natural because it's just environmental exposure. I created an environment when I sneezed that had my pathogen in it and my particles of from my nose. Active immunity means you were exposed and you created your own antibodies. Artificially acquired active immunity is what we typically see with vaccines. Artificially acquired because it's an injection, but you're still creating your own immune response, which is why it's active immunity. Oftentimes vaccines, though, again, reminder, contain weakened or dead pathogens. So you still develop the response, but it wouldn't be a severe response as if you had been infected with the full-blown pathogen. Last, we'll look at the passive ones. Naturally acquired passive immunity. This is best seen. And remember, passive immunity means you're receiving antibodies without exposure to an antigen. So you're not making your antibodies, you're getting them from somewhere. Naturally acquired passive immunity typically comes from breastfeeding. Antibodies are passed from the mother's breast milk to the baby. And then we have artificially acquired passive immunity, artificial being an injection. You can give people antibodies, right? There are many people who donate plasma, and then their plasma can be used, right, to help other people. And that is giving someone antibodies from that plasma through an injection, through an IV, where they're not exposed to the pathogen themselves, so they don't create their own antibodies. They're just simply given them. Here is a nice little table with that. All right, and lastly, we'll look at the hypersensitivities. Yep, hypersensitivities is the same as saying an allergic reaction, an allergy. There are different types of hypersensitivities. And again, this is usually an allergic reaction, a reaction to something, an antigen that is foreign, but generally it's harmless. And so really these hypersensitivities is your body overreacting, being hypersensitive. The first type of hypersensitivity is type 1, okay, immediate reaction. This is when your body produces those IgEs almost immediately. This can lead, in a lot of cases, to anaphylaxis. This is an excessive immune response, okay, which can be life-threatening. You can think about if you're allergic to bee stings or someone who's allergic to peanut butter. If they taste peanut butter or if you get stung by a bee, it's an immediate reaction. That's a type 1 reaction. There's no delay. You immediately start producing those antibodies and you start having that reaction potentially with anaphylaxis. Type 2 is called antibody-dependent cytotoxic reactions. Usually they take a little bit to develop. This type of reaction is what happens when we have a mismatched blood transfusion. Type 3 is immune complex reactions, usually involves a specific type of antibody that usually become autoantibodies, meaning they cause autoimmune disorders like rheumatoid arthritis, where your body attacks its own cells and we don't really know why. And then the last type of hypersensitivity is a delayed reaction, type 4. The best example of this is poison ivy. So typically when you go into poison ivy, you rub your skin against it. You don't have an immediate reaction. You don't start itching. It's not until like days later, even sometimes, or 12 hours that people start to actually have a reaction, and know that they've been through poison ivy. This is a delayed hypersensitivity. It takes a while for those cells to accumulate and cause inflammation. And so it's a delayed response, but it's a hypersensitivity nonetheless. All right, and that is it. All right, please let me know if you guys have any questions, and good luck.